Let's talk about meiosis now. Meiosis is a type of cell division that will produce 4 daughter cells from one parent cell. And meiosis is what we call reduction division. We're reducing the chromosome number. So this typically is used for the production of gametes, and gametes are egg cells or sperm cells. So why do you want to reduce the number of chromosomes in an egg cell or a sperm cell? Well, if you think about it, right, in humans, an egg cell or a sperm cell carries 23 chromosomes. An egg cell will be fertilized by a sperm cell, and once that fertilization happens, the resulting embryo will now have 46 chromosomes. So if you're going through the process of sexual reproduction and you're making gametes and you're combining gametes from 2 different parents, you need for them to have just one set of chromosomes. So the way to get to that one set of chromosomes is the process of meiosis. So we have these terms haploid and diploid. So with meiosis, we start out with diploid parent cells. So a diploid cell refers to the fact that there are 2 copies of each type of chromosome. So an individual is a product of their parent. We all are products of our parents. So all the cells in our body are diploid. They have 2 full sets of chromosomes. One set of chromosome we inherited from our mother, the other set we inherited from our father. So in this image here, we can say this chromosome was inherited from mom, and this chromosome was inherited from dad. They're the same size, and this waistline here is where the centromere is. It's in the same position, and they contain the same genes, let's say. Same thing over here. This one was inherited from mom. This one was inherited from dad. They look the same. Notice the centromere is higher up on these, and they would contain the same genes. And once again, 1 chromosome from mom, 1 chromosome from dad. So this would be a diploid cell. It refers to the fact that you have 2 of every type of chromosome. One inherited from the mother's side and the other one inherited from the father's side. If it's a haploid cell, there is only 1 copy of each type of chromosome. So now we only have 1 of these with the centromere in the middle, 1 with the centromere high up, and 1 with a little bit lower down. And it could have been this is the mother's, or it could have been this is from the father originally. It doesn't matter, but there's just 1 copy of each type of chromosome that is present. So in meiosis, we said it's reduction division, so we're going from a diploid cell, where there are 2 copies of each type of chromosome, to a haploid cell where there's only 1 copy of each type of chromosome. And the reason for doing this is because we want to produce sperm or egg cells that have only 1 copy, so that when a sperm cell meets up with an egg cell, right, if this is our egg cell, and this has 23 chromosomes within it, right, I can draw the 3 there, and we're going to add our sperm cell here. That also has 23 in humans. That's our chromosome number. We have 23 different types of chromosomes. So when these 2 combine together, right, this is going to produce a fertilized egg cell that will now have 46 chromosomes. Right, so each one of these is haploid. We use the letter N to represent a haploid cell and 2N to represent a diploid cell. So our haploid number is 23. So a haploid egg cell meets up with a haploid sperm cell, and that will produce a fertilized egg cell, which is called a zygote, which would be diploid. So as we said, meiosis starts out in a diploid cell. We're using the symbol 2N. We refer to these chromosomes, these sets of chromosomes, as being homologous pairs. So when we looked at the chromosome, we said we've got one that was inherited from the mother and one from the father. So these are what we consider to be homologous because they're the same type of chromosome, as we were mentioned. The position of the centromere is the same, and we should find genes along the length of it that are exactly the same along the length of the chromosome. In this case, we're showing the chromosome and then the sister chromatid, right? So there's the chromosome from mom and then its sister chromatid. Here's the chromosome from dad and its sister chromatid. So these represent what we call homologous pairs or homologous chromosomes. Okay. Now, you may also hear the term non-sister chromatids, right? So this replicated chromosome consists of sister chromatids. Over here, we have another homologous pair in green. So these two together would be a homologous pair. But if you were looking at this one versus this one, right? Or I should circle the whole thing maybe, all right? This one versus this one, we would say these are non-sister chromatids because they're not exactly the same. The genes would be the same, but there could be slightly different information on them. So there could be information on how to produce brown eyes versus how to produce blue eyes. So the gene for eye color would be in the same position on these two, but there could be slightly different information between them. Same thing here. We would say that these are also going to be our non-sister chromatids as well, because they're not going to have the same information. So as we go through meiosis, remember that the DNA is going to be copied during the S phase of interphase. So replication occurs just one time before meiosis, but we actually go through meiosis 2 times. And that's to get it to a haploid condition. So the first time, what we are doing is we're dividing out the homologous chromosomes. And then the second time we go through meiosis, we're dividing out the copies or the sister chromatids. So meiosis 1 consists of the same phases as mitosis. We've got prophase, metaphase, anaphase, and telophase. Telophase is followed by cytokinesis, where we divide up the cell into 2 daughter cells. Then we go through meiosis 2. We go through the same phases of prophase, metaphase, anaphase, telophase, and then we go through cytokinesis to divide our 2 cells into 2 more cells, so we end up with 4 daughter cells. Now notice at the beginning here of meiosis 1, we have 2 sets of chromosomes, or 2 homologous pairs. So we have 1 large set and 1 small set. But there's a total of 1, 2, 3, 4 chromosomes. And by the end of meiosis 1, we have 1 large, 1 small, in each nucleus. So by the end of meiosis 1, we're making haploid cells, 2 haploid daughter cells. Each one of these haploid cells with the 2 chromosomes goes through meiosis 2. We pull the copies apart. We don't count the copies. All right, and then we create 2 new daughter cells, each having 1 large, 1 small chromosome in each. So they're still haploid by the end of meiosis 2. All right, so this is a little bit closer up of meiosis 1. So prophase 1, we would have the nuclear envelope starting to disintegrate. We would have the chromosomes starting to condense and become visible. And we have spindle fibers that are starting to extend outward and make contact with each pair of homologous chromosomes. The spindle fibers are going to pull the homologous chromosomes to the middle of the cell during metaphase, where they're going to line up. And anaphase is when we're going to pull homologous chromosomes to opposite poles of the cell. Then we divide up, I should say, we package up those chromosomes into their own nuclei. And then this is followed by cytokinesis, which divides the cell up into 2 daughter cells. So once again, we're going from 1, 2, 3, 4 chromosomes to 1, 2. So instead of having 2 copies of the large chromosome, we have 1 copy. Instead of 2 copies of the small chromosome, we have 1 copy in each of the cells. Notice they don't look the same, the 2 nuclei. One has the large purple, this one has the large red. So this could have originally been inherited from the mom, and this could have originally been inherited from the dad. So there could be slight differences in how that information is encoded between these 2 chromosomes. Meiosis II actually follows the same steps as mitosis, right? So there's not a lot of difference here. The concept is that we are going to pull the sister chromatids apart. So prophase 2, the nuclear envelope will disintegrate, the chromosomes will condense. If they were uncondensed in between, spindle fibers will reach out and make contact with each sister chromatid and pull them so that they're lined up in the middle of the cell during metaphase. So there's no more homologous chromosomes during meiosis II. We just have the individual replicated chromosomes. So spindle fibers should be attached to each sister chromatid, so that during anaphase, when the spindle fibers contract, they're pulling sister chromatids to opposite poles of the cell. And then each side of that cell with those chromosomes are going to be packaged up in their own nuclei, and cytokinesis will divide it into 2 daughter cells. So since we're following 2 cells through meiosis II, we now have 4 daughter cells that are produced. Now once again, look at what's occurring inside the nucleus. We have a large red chromosome, large red here, but we have large purple chromosome in these 2 daughters. We have the small purple here, but we have the small red down here of the chromosome. So these daughter cells are not genetically identical to each other. They're a little bit different from each other. So every egg cell or sperm cell that is produced can be different from other egg cells and sperm cells produced by the same individual.